In the last video, we looked at how we go about classifying monosaccharides based on how many carbon atoms they have and whether they have an aldehyde or ketone functional group. In this video, we are going to dive into another key consideration with monosaccharides, and that is their stereochemistry. In the case of monosaccharides, the majority of these have one or more chiral centers, and in fact, in most cases, the molecules have multiple chiral centers. And what that means is that there are many different stereoisomers that can exist of these molecules. Different stereoisomers can have very different biological effects, and hence we need to be able to readily evaluate the stereochemistry of monosaccharides. Let's take a closer look. Glucose, arguably the most well-known of the monosaccharides because of its key role as an energy source in humans and other organisms, is a key example of how monosaccharides have interesting stereochemistry. If we take a look at glucose, here's the line angle formula of the acyclic form of glucose, where we have our chain of carbon atoms here. If we take a look at this chain of carbon atoms, we can recognize chiral centers here, 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 and here. You could use the skills that you learned in prior videos to go through and assign each of those chiral carbons as R or S. We also have this ball and stick model to help us see some of the three-dimensional features of glucose. And then finally, here at the right, we have one way of going about drawing a Fischer projection of glucose, where remember in the Fischer projections, we draw the carbon chain vertically, and by vertically, what I'm referring to is going from top to bottom here, where the top carbon of that chain represents the aldehyde group if it's an aldose. If it's a ketose, the ketone group would be oriented so it's as close to the top end as possible. And then going down the chain, each of the intersections represented here, 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 and here represent a chiral carbon. So in the case of glucose, we recognize 4 chiral carbons here, 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 and here. This carbon is not chiral because it has 2 identical hydrogen atoms bonded to it, and therefore there's some unique stereochemistry here. In order for this molecule to be referred to as glucose, it absolutely has to have this orientation at each of the chiral carbons. In other words, at each of the chiral carbons, what we have to find is that coming from the aldehyde group at the top, the OH group has to be on the right at this second carbon of the chain, OH group on the left at the third carbon, OH group on the right at the fourth, OH group on the right at the fifth. If there's any other orientation at these chiral carbons, then the molecule is no longer this stereoisomer of glucose, but instead something else that would have completely different biological properties in terms of how it's metabolized and how it is perceived as sweet or not sweet by the taste receptors. And as a reminder, when we are talking about Fissure projections, the way that it's drawn here in our pre-made schematic is a bit more spelled out than you may at other times see Fissure projections spelled out. It would also be perfectly accurate to write out the aldehyde group as just CHO, and then coming down the chain, drawing the chain vertically, just making 4 intersections of lines here to represent the 4 chiral carbons, where it's implied that the intersection of the lines here, 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 and here represent carbon atoms. And then at the terminal end, the bottom end here, plugging in CH2OH. Now at each of those positions along the chain, it's essential that we correctly orient the hydroxy group at each of the chiral carbons. If it's not a chiral carbon, then it doesn't matter whether the OH group points to the left or right. It would be the same exact molecule one way or the other. So down here at the CH2OH, I could have put the CH2OH off to the left. That would have been fine as well. But at each of these chiral carbons, that's where we have to pay close attention to the orientation of the H relative to the OH at each of those. Otherwise, we'll get the stereochemistry wrong, and hence we will not have the same molecule. We will have very different molecules and very different biological effects as a result. So I've redrawn this Fischer projection in a more condensed manner over here, but these two structures are completely equivalent in every way, including the stereochemistry of the molecule. Now, what can we discern from the stereochemistry here, and how can we look at different molecules and relate those based on the stereochemistry? The first thing that we will discuss is how to determine whether a sugar is a D sugar or an L sugar. So how to determine D versus L configuration or stereochemistry of the sugar. Commonly, the D configuration is the configuration that is most well known and most widely used biologically. So when we think about the glucose that is present within our bodies, that particular stereoisomer is D-glucose. The way that we recognize D-glucose versus L-glucose is the D designation at the start of any sugar's name is going to indicate that the final chiral carbon has the hydroxy group on the right. So I'm going to go ahead and plug in here that D means that the final chiral carbon, meaning the one closest to the bottom of the molecule in the Fischer projection, has the OH group on the right. In other words, having the OH group on the right at this position will cause the molecule to rotate light in a clockwise direction, because that's what D refers to as that polarized light is rotated by the molecule in a clockwise direction. So in other words, if you filled a tube with this sugar solubilized with water, put it into a special instrument called a polarimeter, it would cause the light in that polarimeter to rotate toward the right. And that is what we refer to as D. And that's going to be true of any sugar that we describe as a D sugar, where this OH group at the final chiral carbon is on the right. That is why we would refer to this as the D configuration of this particular molecule. On the other hand, the L configuration of glucose or any other sugar would refer to the situation where the final chiral carbon, that is the chiral carbon closest to the bottom, has the OH group, instead of being on the right, on the left. That would enable the molecule to be referred to as the L configuration. So in other words, if this OH group that I'm highlighting here in our structure on the far right, if this OH group were not on the right, but instead it were over here on the left, the molecule would instead cause light to rotate toward the left counterclockwise, and the molecule would be referred to as the L configuration. Now let's tackle the task of determining the relationship between some different representative sugar molecules. For this exercise, I'm going to refer back to this flowchart that you can find on Canvas with the materials for this chapter, and this flowchart was also discussed in detail back in video number 349. We are going to use this flowchart to evaluate the relationship between pairs of monosaccharides. So for example, what I want us to do here is specify the relationship between the two sugar molecules that I'm going to draw here. It's important to be able to recognize the relationship between these monosaccharides because the differences in structure will lead to differences in biological function. So let's go ahead and draw out a couple of structures here. Going to go ahead and draw these out as Fissure projections, which are a convenient way of representing the stereochemistry of monosaccharide molecules. Fissure projections were developed specifically for that purpose. And the two structures that we're going to compare are these two here. And here. So I'm drawing these out. Like so. And we're going to look at the relationship between the two sugar molecules here. So taking a look at the two molecules, the first thing that we need to do to make sure that this question isn't just out of left field is establish that the two molecules are actually indeed isomers of some sort. Isomers, recall, are molecules that have the same number and type of atom, meaning the same molecular formula, such as C3H8O, but have a difference in structure of some sort. In both of these molecules, we can establish that they have to be isomers of some sort because in both cases, the formula is C5H10O5. And this formula very nicely fits our prototypical formula of monosaccharides because it has that formula CnH2nOn in both cases. So looking at these two structures, we established they have to be isomers of some sort because they have the same molecular formula, but they have some sort of structural difference. So going down the flowchart here, the first distinction that we can make is to establish whether the two structures are related to one another as so-called constitutional isomers or whether they are stereoisomers. Constitutional isomers have to have a difference in the skeleton where there's a different connection between atoms such as different branches along the chain or branches at different positions along the chain or different functional groups, like one of them has an aldehyde and the other one has a ketone group. As a result, the structures would ultimately have different IUPAC names, and we could name these using the IUPAC designation if we wanted to, and that would be one way to determine whether they are constitutional isomers or not, as they would have different IUPAC names. Stereoisomers, on the other hand, will have the same connections between atoms, meaning they would have the same IUPAC name that designates the branches and the parent, but they have a different three-dimensional shape, such as at one or more of the chiral centers, the R configuration has switched to S or vice versa. Now within the bucket of stereoisomers, we take that a step further down the chain here to delineate whether molecules are related as so-called enantiomers or as diastereomers. Enantiomers are going to be molecules that are non-superimposable mirror images, like your left and right hand. The way that you can recognize an enantiomer is if you look at two molecules and you recognize that they are mirror images of one another and they have a chiral center or more than one chiral center, then the two structures are enantiomers. Another way to recognize it is that every single chiral center has the opposite configuration as we compare the two structures. Not just some of the chiral centers have switched, but all of them have gone from R to S, or in the Fissure projection, the way you could think of that is all of them have gone from left to right in terms of whether the hydroxy group is to the left of the chiral carbon or to the right of the chiral carbon. If every single position that's a chiral carbon has switched, then the two structures are related as enantiomers. Diastereomers is everything else. So diastereomers are all of the molecules where some, but not all, of the chiral centers have switched configuration from R to S, or in the case of the way we've drawn these, from left to right is a way that we could look at it, where the OH groups on some of the chiral centers have switched from right to left, but not every single position. So that's how we tell enantiomers from diastereomers apart. Enantiomers are mirror images because every single chiral center has inverted its configuration. Diastereomers are where some of them have, but not all of them have. So looking at the relationship between these two sugar molecules, as we take a look at these, we can rule out constitutional isomer as a possibility here. And the reason why is as we look at the two, they would have the same IUPAC name to specify the parent and the branches, because in each case, they start with an aldehyde group. They both have a total of 5 carbons in their longest carbon chain, and at every position, they have a hydroxy group along the chain here from carbon 2 to carbon 5. And therefore, they would have the same IUPAC name with the exception of possible designations of stereochemistry. So they have the same branches in the same spots and the same parent name. So therefore, the other option here is stereoisomers. And we can't just say they're stereoisomers. We need to be more specific and distinguish between diastereomer and enantiomer. So we have to ask ourselves, are the two structures mirror images at the chiral centers, or is there something about it that's not a mirror image? So look at the relationship between these two sugars. I think the most straightforward way to recognize they're non-superimposable mirror images is ask yourself at every single chiral center, has the orientation of the OH group switched from left to right? So taking a look at each of the chiral centers and comparing apples to apples, we're comparing this chiral center to this one because those are in the equivalent position in each case. And taking a look at that, indeed, what started off on the right here was the OH group, and now it's on the left. So we would say that that particular stereocenter inverted. So we inverted the chiral center, in other words, because it went from being on the right to being on the left, making this position a mirror image of itself. We can look at the second position because in order for it to be enantiomers, we have to have switched each and every one of those positions. So here we have the OH group on the right. Here the OH group is on the left. And so this would be another spot where the stereo center, or chiral center as you could synonymously call it, has inverted. Come to the third spot in the chain, or the third chiral center, I should say. It's really the fourth carbon of the chain, but third chiral center, here versus here. Comparing those two here with the equivalent spot over here, you see that the OH group is on the right here and on the left for this structure. Therefore, that has inverted as well. That represents 3 of 3 chiral centers here, here, and here. All 3 of those have inverted. Therefore, the 2 structures are the perfect mirror image of one another. All the chiral centers have inverted. And since all the chiral centers have inverted, we describe these molecules as being related as enantiomers. So with the information that I have provided here in this flowchart, we should be able to apply this to compare the structures of any 2 sugar molecules and describe how they are related, where the possible relationships are that they aren't isomers of any sort, meaning they have different molecular formulas, they're constitutional isomers, or they're stereoisomers, which when we talk about stereoisomers, we need to be prepared to describe stereoisomers as either diastereomers or enantiomers. So the 3 key terms here that we will use to describe the relationship between 2 molecules that have the same formula is constitutional isomer, diastereomer, or enantiomer when we're referring to the relationships between different sugar molecules that have the same molecular formula. In the next video, we are going to dig into some additional problem solving to apply this terminology toward identifying the relationship between additional sugar molecules.